and welcome back to my YouTube channel. This is the part 3 of chapter Circulatory System, and today we are going to study about a cellular component found in blood, that is red blood cells, also known as erythrocyte. Now the red blood cell is red in color due to the presence of a pigment, that is hemoglobin. Hemoglobin is a respiratory pigment that binds with oxygen to form oxyhemoglobin, which is transported to different parts of the body wherever it is required, and there oxygen dissociates. Now the structure of red blood cell is biconcave, that means it has two concave surfaces, one at the front and one at the back, and it is thick towards the periphery. This type of structure of RBC makes it very efficient for absorbing oxygen due to the presence of larger surface area. The smaller size enables it to move through the capillaries, that is 7 micrometer in diameter and 2 micrometer in thickness. In males, there are about 5 million RBCs found, and in females, it is about 4.5 million. Now, the pigment that is found in red blood cell is hemoglobin, which is made up of two parts. The first part is hemin, that is iron containing part, and the second part is the protein part or the globin part. This hemoglobin combines with oxygen to form oxyhemoglobin, which dissociate into oxygen and hemoglobin wherever oxygen is required in different parts of the body. Hemoglobin carries small amount of carbon dioxide in the form of carbaminohemoglobin, although we know that most of the carbon dioxide in our blood is carried by plasma in the form of carbonates. Carbon monoxide poisoning happens when a person sleeps in a poorly ventilated room in the presence of burning furnace, which burns in the insufficient supply of oxygen, and therefore it leads to the formation of carbon monoxide, which has strong affinity for hemoglobin. It binds with hemoglobin to form a carboxyhemoglobin, which is a very stable compound, and therefore it cuts down the capacity of hemoglobin to bind with oxygen, leading to carbon monoxide poisoning, ultimately death. Production and destruction of RBC. The RBCs are produced in the marrow of long bones, especially in the case of ribs, breastbone, ilium and hip girdle. In embryo, they are produced in liver and spleen. The mature RBCs lack nuclei, but they have one when they were produced. As they mature, the nuclei is lost, they become enucleated. This increases the surface area by volume ratio for absorbing more oxygen. There are no membrane-bound organelles present in RBCs such as mitochondria, so that the RBCs may themselves not utilize the oxygen and thus fulfill their purpose properly. There are no endoplasmic reticulum present in the case of RBCs in order to provide them enormous flexibility to move through the capillaries easily. The average lifespan of RBC is about 120 days. The old RBCs get destroyed in liver, spleen and bone marrow. With this, we are ending our discussion based on the cellular component RBC. Hope you are liking my lectures. Do like and subscribe my channel. Thank you.